All right, so now we're going to get into each of the different metabolic pathways. The first is glycolysis. Glycolysis occurs in pretty much all organisms. In eukaryotic organisms and in prokaryotic organisms, glycolysis occurs in the cytoplasm of the cell. This is an anaerobic process, so we're not using any oxygen at all. And the whole function of glycolysis is to take a glucose molecule and convert it into 2-pyruvate molecules. When we're doing this, we're going to have to invest a little bit of ATP. And so here we see ATP being invested. ATP is invested. We add actually a total of 2 ATP to invest. And that activates the glucose molecule. Glucose is then going to be converted into multiple different molecules, so these are the intermediates, through different enzymatic reactions. In the process, we break glucose into 2,3 carbon molecules. We oxidize the glucose, so here we have NADH being produced, and we have substrate level phosphorylation occurring. The end result then, we're going to have these 2 pyruvates, which are 3 carbon molecules. We're going to have a total of 4 ATP produced, but since we use 2 ATP at the beginning to activate our glucose, we only gain 2 ATP net benefited. And then we have these NADH that are produced that are going to move directly to the electron transport chain if we are going into aerobic respiration. If we're not, then the NADH are going to have to be oxidized so they can keep the glycolytic pathway running. So let's move into the aerobic respiration pathway. From the glycolytic pathway or the glycolytic step, we're going to move into the transition step. So this occurs in bacteria in the plasma membrane of the cell, but in eukaryotes, this occurs in the mitochondria. And so we started with that pyruvate that was produced here. The pyruvate is going to be oxidized, and we're going to lose carbon dioxide or a carbon from it. Oxygen is going to pick up the carbon, producing carbon dioxide as a waste product. NAD is going to be reduced to NADH. And the molecule that's left is called an acetyl group that will be carried by coenzyme A. Coenzyme A is another carrier, another taxi, and its only job is to take acetyl groups to the citric acid cycle. So we move into the citric acid cycle. Again, this is going to occur in the mitochondria of a eukaryote and in the cell membrane of a prokaryotic organism. In the citric acid cycle, we are going to completely oxidize the entire molecule, the entire rest of the glucose molecule. And so we start with, this is a cycle, so we start with this 4 carbon molecule called oxaloacetate. The acetyl group is dropped off and binds to this 4 carbon molecule, producing a 6 carbon citric acid that is going to lose all of the carbons and be oxidized completely back to the 4 carbon molecule oxaloacetate. We have NAD and FAD waiting to pick up all the hydrogens and electrons that as the molecule is oxidized, we have oxygen available to pick up the carbons. And in this process, we produce a little bit of ATP through substrate level phosphorylation. So we're going to watch this video.
During glycolysis, glucose is broken down to pyruvate. A 2-carbon fragment of pyruvate is used to form acetyl-CoA. The acetyl-CoA enters the Krebs cycle, which occurs in the mitochondrion. During the conversion of pyruvate to acetyl-CoA, carbon dioxide, CO2, is produced and a molecule of NADH is formed. The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA 6-carbon molecule.
Sorry about that. I'm going to back it up a little. So
the CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
All right, so that's that Krebs or citric acid cycle. I didn't mention, so apologies. Citric acid cycle is named for the production of the citric acid. And it's also known as Krebs cycle because the individual who found it was Krebs. That was his name. This is the video you just watched, but it doesn't work, so I just went to the video, sorry. So the next thing we're going to talk about is the chemiosmotic theory or oxidative phosphorylation using the electron transport chain. We've produced a bunch of reducing power in the form of NADH from glycolysis, transition, and citric acid cycle. We've produced some FADH from citric acid cycle, and these are all going to be dropped off at the electron transport chain. So the electron transport chain itself was proposed, and then the research was supported. In 1961, Peter Mitchell proposed this theory of producing a lot of energy, and it's now called the chemiosmotic theory. So initially, nobody believed it, and so he did a lot of work on his own, and he had to fund most of his research. But about close to 20 years later, his research was fully supported, and he actually won a Nobel Prize. And so this is Peter Mitchell. So what is the electron transport chain or chemiosmosis? Basically, you're going to take high energy electrons from the oxidation of glucose, and you're going to move them down an energy gradient. And as we move them to lower energy levels, we're going to cause them to pass the protons, or to pump the protons across. So we're pumping protons against their concentration gradient, or against their, up a concentration gradient. So normally molecules want to flow down their concentration gradient from higher to lower. We're pushing them against that gradient. So we're going to produce a very high energy gradient of a bunch of protons or hydrogen ions, they're the same thing, that are going to be on one side of a membrane, and they're going to want to move from higher to lower concentration. So in prokaryotic organisms, this occurs in the plasma membrane. In eukaryotes, this occurs in the cristae of the mitochondria, the inner membrane. And so this is what it looks like. Here we have the different protein pumps. As hydrogens and electrons are dropped off, the electrons move from one carrier to the next, and that pushes the hydrogens to the opposite side of the membrane. So we have this very, very high gradient of protons in this region and fewer protons down here. The hydrogen ions can only move through this molecule, this protein complex called ATP synthase. As these hydrogens flow through, they activate the molecule and ATP is formed. Depending on if you are NADH or FADH dropping off your electrons will determine the amount of energy. NADH produces 3 ATP per hydrogen, while FADH that drop their electrons off further down, so they don't have as much time for those for the energy to be released, only gain 2 ATP per hydrogens. And so the end result is you're going to produce about 30 ATP from the oxidation of the NADH back to NAD, and 4 ATP from the oxidation of the FADH back to FAD. Some bacteria can use an anaerobic electron transport chain. So when they are using an anaerobic electron transport chain, they can't use oxygen as their final electron acceptor. Oxygen is the final electron acceptor of aerobic respiration. But in anaerobic mechanisms, you can't use oxygen, it's anaerobic. So they're going to use something else, such as sulfate or nitrate or potentially an organic molecule to use as the final electron acceptor of anaerobic respiration. In this mechanism, you're not going to produce as much ATP either. And so anaerobic respiration, though it uses a modified citric acid cycle, it uses an electron transport chain. It doesn't produce much energy compared to aerobic mechanisms. So the amount of ATP produced then is roughly 38 ATP. You have a total of 34 ATP in the oxidative phosphorylation, but you also have 2 ATP produced in glycolysis and 2 ATP produced in citric acid cycle. And so that gives you a total of 38 ATP. So here is the complete breakdown of glucose, where all the energy is formed. And then again, you can see the maximum amount. If you are 38 for 1 molecule of glucose, but if you're a eukaryotic organism, then you get a little bit less ATP because we're pushing our pyruvate, our NADH, into the mitochondria to drop off the hydrogens and electrons, and so we don't get as much energy from that. The next mechanism is fermentation. Fermentation is used when we can't respire. So humans use fermentation when we are, or when we don't have enough oxygen for aerobic respiration. This is a mechanism that allows us to produce energy really rapidly, not a lot though, just a small amount. And so think about when you're running or when you're at the gym lifting weights, the burn that you get in your muscles, that is because your muscles are fermenting, because you're using more energy than can be used just by aerobic respiration means. E. coli use aerobic respiration when they have oxygen. That's their preferred means of energy production. They can use anaerobic respiration, and they use fermentation. Some bacteria only use fermentation as their mechanisms of producing energy. So fermentation, and I think, oh, right here, sorry, I have it right here. Fermentation is just using that glycolytic pathway, and then we're going to take that pyruvate and we're going to reduce it. And when we reduce it, we reproduce the NAD, which can then be used in the glycolytic pathway to break down another glucose. And the end product is going to be dependent on what type of organisms. So humans and lactic acid producers, so I say humans because we do produce lactic acid as well, but so does like lactococcus, produces lactate. And so here we have pyruvate is reduced to lactate, NADH is oxidized back to NAD. Here we have pyruvate releasing a carbon dioxide and being reduced to acetaldehyde, or so, no, okay, let me say that again. Pyruvate releasing a carbon dioxide, forming acetaldehyde, which is then reduced to ethanol. So these are all mechanisms or all end products, I should say, of the fermentation process. And a lot of these are very important to us because it's how we produce some of the foods that we eat. And some of these are important because they help us to identify our bacterial species. So we don't have to use glucose. Glucose just tends to be our favorite molecule for breaking down because glucose produces energy very easily. But we can use other molecules. We can use fats, we can use proteins, but when we do, we have to modify those molecules first before we can break them down, which is why we oftentimes use just glucose. If it's available, we like to use glucose. It's our favorite fuel in our bloodborne substrate pool. And if you don't know where that came from, listen to the glycolysis video that I have. I'm going to stop here, and then we're going to go into photosynthesis next.